Okay, I'm going to go ahead and get started. The content for today is in Module 4. There's a PowerPoint in the lecture content. That's really the only content there so far. So Tuesday lecture has a link to download the PowerPoint slides. Plan for today is to, well, the overview, or the big idea for this week, translating biological questions into computational solutions, or at least computational questions. The specific example is taken from the first chapter of this book. And there's a link there to the textbook website. This is a few years old now, but that doesn't really matter, just in the sense of this is a third edition. Not sure what edition they're on now, but they made a whole series of lectures to go with it. And the Rosalind homework platform was originally designed to go along with this textbook. So the problems are, the Rosalind platform has a lot more questions than just what's in the textbook, but for this module, they're plucked out of the first chapter of this book. And I guess backing up a step, any questions? I come in ready to jump into this week because I'm excited to talk about the next thing, and I forget about what just happened, which was last night for some of you. I haven't looked over the homework. I've pulled the Rosalind grades. It looked like almost all the problems were completed by everyone. And so, like, take a breath for me. And are there any questions about any of that stuff from last week? Okay. Yeah. So if you get code that you don't recognize from ChatGPT, lots of things you could do with it. You can ask it to build on what you've done before. You can ask it to explain. So that's another thing that we're going to get into today. So the last two slides in this slide deck are how to use ChatGPT as a tutor. So there's a sort of 6 steps that you should use on every Rosalind problem so that hopefully it helps you understand. Because even when I ask ChatGPT, and I would say I'm no super programmer in Python, but I know Python. Occasionally I get a function that I didn't know existed. And so, yeah, that opportunity to learn some new coding method or new algorithm. In this class, you guys, as sort of, well, many of you new to Python, there's going to be lots of things that you don't recognize. And so that's, again, what hopefully the last 2 slides will at least give you a prompting strategy to help you learn from ChatGPT. Learn to learn. That's the good question. That's also why I did the git commit route, so that I can look at how you guys progress. Especially at the end of the semester, I'll dig into those in some detail to help inform strategies moving forward for how to teach this stuff in the age of AI.
When we're using ChatGPT and it gives us a code that, like, we can see that it's not like what exactly we want, and then we try again, should we leave the first code that we have that we're not going to use and, like, just keep that in our gigantic folder, or should we remove the ones that we're not going to use?
Yeah, it's a tough one to answer because it's on a continuum, right? So some stuff you're just going to delete and forget you ever did that prompt and that answer ever existed. In which case, um, yeah, fine. But then you have the whole continuum to the other end where it gave you exactly what you wanted, and that's what you're using. In the middle somewhere are half correct, where it's just it's valuable in the sense that it was instructive about how you get to the end, and that is stuff that I would like to see in there. So how, because that tells me, like I was saying, at the end of the semester, I'll dig into the uh, commit histories and see how did you guys learn to solve a certain problem. Other questions? And I think as a habit also that strategy is valuable, because then you can go back, because you put some computational project aside for a few months and then come back to it. And if you haven't commented it well, good luck. You're starting from scratch again. Whereas if you have a commit history, then you can sort of track your own logic through progressing to wherever it is at the end. Okay. All right. So the problem, or the uh, biological question that we're going to address, is about chromosomal replication. So I'm not, if you have a question, definitely stop me. I think we have more than half biologists in here, but certainly for the online people who are more disproportionately data science, I have some of the biology in the slides, right? So this is a bacterial chromosome, and it's showing replication. So when the cell divides, it needs to replicate its genome. And so here you've got the start of replication, and then as it proceeds, the two strands of DNA unwind, and both strands are being replicated in opposite directions. So you can sort of visualize this circle is being pulled apart from one side, and in the end, you're going to have two circles, to like two whole genomes, and then the cell can go ahead and divide. And so the biological question is, where does this process start? Or what we'll get to is, how does the cell know where to start replicating its DNA? It's not random. It always starts in the same place. That place is called the ORIC, the origin of chromosomal replication. We ask this question a little bit more formally, and it's not an accident the way this is formatted, because that starts to look like a Rosalind problem, right? Given something, return something. So in the biological sense, the question is, where is ORIC in a genome? So if we're given a genome, we want to return the location of the ORIC in that genome. That's the biological question. So for the wet lab people, so people who come from biology background maybe, is that well enough well enough defined for you to approach experimentally? Like, just thinking as a biologist, could you answer that question? If so, how? Feel free to talk amongst yourselves. What might you do as a wet lab biologist to answer that question? Yep.
Should we use like PCR and RNA primers to kind of trial and error and see if that works?
Could you do some kind of PCR to find out? Um, maybe. I actually I don't think so. It's a good stab at it though. Any other ideas? You yeah. The reason I'm a little hesitant is there might be some quantitative PCR that you might be able to do if you could get it in like the windows to see it, like you could potentially capture duplicated versus non-duplicated regions because there will be more copies of it. So if you're doing quantitative PCR and you did it, a lot of it, you might be able to see, well, this region is more often, I'm catching it in a duplicated state. And so that would tell you it's early in the replication cycle. Um, so maybe. Any other guesses that, or just no? Like, I don't think he could do it. Yeah, that's the answer for like developmental biology as a discipline. Mutate the crap out of it and see what gives you the phenotype, like what can't replicate anymore, and then go find out, okay, what got mutated in that thing. What about computationally? You think you could write some code to figure that out? So you're already nodding your head. What would you do?
So, I map out the gene and
But we don't know what the gene is.
it, oh,
We know there is one because we know replication always starts in the same spot, but we don't know where that spot is. Yes, we'll pretend we have the genome.
Use information derived from other experiments where you found, or other species where you found the origin of the OREC,
Yep.
and get that fragment, just search for it, the fragment of the experiment, and see if it comes up.
Yeah, life is easier the more we know, right? So if you know what it is in some species, you can go look for that sequence in the next one. The problem is for this, it doesn't work very well. So like we'll get there in a minute, but yeah. So pattern matching.
so the origin, it's not the same as every genome, right? So
The
we would need,
sequence is not the same. There is an ORIC in every genome, bacterial genome.
right, but it wouldn't be the same as another a different, like you can't like just isolate it to find one ORIC.
Some are the same and some are not.
Okay. But the point is you can't isolate it.
Right, right. Okay, so we got the biology answer. We can do a screen, look for things that don't replicate, right? So this is an example where this plate on the bottom right, obviously those colonies are mostly not replicating. And so you could go back and see, okay, what got mutated in the line we plated on this plate. For the bioinformatician, this is not like all those solutions are ones that we're going to go after, but this is not really a well-formed computational question because looking for a pattern, that's a next level question, but that's sort of where we're headed. How do we turn this into a computational question? So starting to define the computational question, we can ask, well, how does the cell know where to start? And I mean, what you were starting to get at with the pattern matching, or whether it is the same in different species, is there some signal Like, is there a consistent DNA signal that says start here? Okay, and the translation to well-known computer puzzles, challenges, are hidden messages. Just as a kind of interesting aside, do you know, this will hopefully become relevant in a minute, what Darwin was reading that gave him his eureka moment for evolution by natural selection? No, that maybe informative, but not sort of the eureka moment that gave him the not the things evolved, but the natural selection specifically. It's an economics textbook. Thomas Malthus Essays on Principles of Population, right? So the relevance here is that it's interesting to me, and I think informative, to know that lots of eureka moments come by reading widely. And so this connection between what's the signal in DNA and hidden message problems in computer science doesn't get made unless there are computer scientists interested in biology questions reading some microbiology literature, or at least collaborating over coffee, right? That sort of cross-pollination is how bioinformatics became a field, right? I have this biology problem and a ton of data, but I really don't know how to address it. We'll get to in the next, maybe next module, module 5, BLAST. Like, how do you search millions and millions of sequences really fast to find pattern matching? That was a really hard biological problem in the late 80s, early 90s, that computer science came along and said, hey, we've got this set of algorithms that searches really fast heuristically. I think you could search DNA that way. And, sort of revolutionized the early genomics field, being able to search massive database of sequence. So that's the cross-pollination here of hidden message problems are a classic bridge between sort of human logic puzzles and computer algorithms. So ciphers and encryption software all sort of go back to this, try to figure out what the hidden message is. This is from the textbook example. They go back to the gold bug essay, Edgar Allan Poe, published in 1843. This is one of the classic examples of a ciphertext, and then figuring out what is the cipher, that is the algorithm that translates. And they do it by frequency analysis. So the only hint you're given is that this is in English, okay? And so knowing that this is in English, you can go through and say, well, we know that certain letters are overrepresented in the English language, like E's, like anybody watch Wheel of Fortune, right? There's a reason they give you certain letters in the sort of final round, because those are super common and everybody would guess them. So just knowing that this is English, you can break this text. If you're curious, you can ask ChatGPT to do this cipher, but you have to tell it not to just look it up, because it's so common on the internet, it will just give you the answer without actually solving the cipher. So we're getting closer to defining a computational problem. So the hidden message problem now is, how do you find a message in the string? So if we have a string that's text representing the ORIC region, it's a DNA string, return a hidden message. Well, what is a hidden message in the context of genome sequence? Well, like I said, in the gold bug context, it used frequency analysis. So we can apply a similar strategy to this genome context, what's a hidden message, are certain DNA patterns more frequent than you would expect by chance. Okay. So again, just for fun, already got ahead of myself there. Some steps. It's kind of interesting to see ChatGPT work through that solution. But that's effectively what it does is what we're going to do, taking this frequency analysis approach. So do certain k-mers, that's just the shorthand for a string of length k, appear surprisingly frequently in small regions of the genome. So we're going to start with small regions of the genome, then we'll expand to looking at whole genomes in a minute. So for example, if you've got a 5-mer, right? So the k-mer here, k is 5. In this string, for that 5-mer to happen 3 times, sort of surprisingly frequent, right? So that's what we're talking about. Is there a pattern that occurs more frequently than you would expect? And just in case it's not clear, right? We've got 2 levels of this question. How does the cell know where to start? That's hidden messages. And then what is a hidden message? That's going to be what's the most frequent k-mer? What's the most frequent pattern that's in that string? So given the region, what's the most common k-mer? And for this simple case, we're going to be given what the k is that we're going to look for. We're not going to say it can be any k, any length k. So in this example, right, our pattern is AATTT. So the k-mer length is 5, and then our text or our genome is that string across the bottom there. Okay, so now, whoops, that's the wrong. We have a computational question, right? So we can, and this is back to if we knew what to look for, and then to start out with, we're going to say this is what you should look for. This is the pattern. You can slide along that text and report how many there are, and then find out which of the patterns is the most common. So does that make sense to the biologist? So like flip that around, right? We're having coffee talking about our biology question and going back and forth with our computer science friends. Does that make any sense? If I was to tell you the most frequent k-mer in the region, would that mean anything to you? And the biologist, knowing something about transcription factors and how the chemistry, the biology of replication works, would say, yeah, we know that there's a DNA polymerase that sort of initiates this process. And so it must have some kind of recognition site. We know transcription factor binding sites are pretty small, maybe not a 5-mer, but a 9-mer is not unreasonable. So this DNA-A is the polymerase that does it in most bacteria. We know that the DNA binding site is roughly 9 nucleotides long, and that is what initiates replication. So yes, to at least first approximation, this approach is potentially going to give us a meaningful biological answer. It's going to tell us the binding site for DNA-A. So we're going to come back and solve this Rosalind problem at the end, but I really want to finish working through the biology of how to approach the rest of, like, the problem is not solved yet. But number 10 is going to introduce the sliding window algorithm. In some of the Rosalind, you see it gives you pseudocode, and I'm going to show you at the end how to ask ChatGPT to give you pseudocode, which isn't as cryptic as this. But the book that this comes from uses this shorthand, like count with this back arrow and a 0. That means assign a variable named count the value of 0. And then the shorthand where it looks like the absolute value of something, like it's got the pipe and then a word and then a pipe, that means the length of that thing. So shorthand for, right here, it says I equals 0 to the length of text minus the length of pattern. That's the English translation of that line. And in case I forgot to mention it, I left that code in there. So you'll see this, I'm sure, in the solution for this text. So this len is length of, and then whatever variable's in the parentheses, it's going to give you the length of that variable. The next step in that problem, so number 10 will compute the number of times the pattern appears. So if you go back like here, solving this problem, it would say that given that pattern, it happens 3 times. The next step is to find the most frequent words. So what you'll have to do is go along the genome, find all of the k-mers that are 5 base pairs long, okay, and then count them. So then tell me which k-mers were the most frequent. So rather than given a pattern, now what are all the possible patterns that are in there and which one is most frequent? So we'll come back to those, see how much time we have at the end. We may solve 10 and 11. So I kind of glossed over the frequency issue of how common, like I showed that 5-mer and that it happens 3 times in that short string of DNA, but is that really a surprising number of times? What's the expectation if the DNA sequence is just random? There's actually no hidden message in there. So that gets us to this issue of what's the probability of seeing a given k-mer in the text at all. So in this example, so this is the ORIC from Vibrio cholera. This is the bacteria that causes cholera. And these 2 k-mers, 9-mers, happen 3 times each in this little region. Is that surprisingly common? So how do we address that question? Like when do we start to say this frequency is maybe that's a hidden message? There are 2 kinds of probabilistic models. One is the multinomial model. This is the simplest model, and the one that we'll sort of apply as our back of the envelope calculation for how frequent do we think something ought to be. It assumes that all nucleotides are independent and identically distributed. So that's just what this is showing right here. So what's the probability of seeing an A, a C, a G, or a T? Well, if we just take a naive assumption that they have uniform distribution, there are 4 of them, so the probability of seeing any one of them is 1/4. Okay. That really all of this whole slide is saying, except that, like, if you want to say what's the probability of seeing 4 specific nucleotides in a row, in this case ACGT, you just multiply 0.25 times 0.25 times 0.2. Like the probability of seeing each one is independent of what came before it. So you just multiply its probability over and over again. Okay. And so the probability of ACGT is exactly the same as GGTT, same as CCCC. Okay. And for the biologist, maybe you think, oh, that's not very realistic, but it's a null model. It is a naive model. An improvement on that model would be, okay, scan your genome and say, what's the actual frequency of A, C, Gs, and Ts in there? So if we have a really AT-rich genome, you might want to say, well, the probability of getting an A or a T is a little bit higher than a G or a C. You could make that modification if you wanted to. and it would give you a better expectation. Okay, I'm seeing some glazed over looks, like I lost you. So pretend your genome is all A's, super extreme, right? There, in that case, you wouldn't want to have your null expectation to be like CCCC should have a 0 expectation, okay? And so you could modify the expectation based on the nucleotide frequencies in the genome. That's all we're saying. The naive expectation, we're just going to go in like we don't know anything, and on average, it's a roughly good approximation that we're going to use 1/4 for all of them. So that's multinomial models. The other main class of models are Markov models, and these can get really sophisticated. And the idea behind them is this, that you want to model the probability of getting this pattern of DNA based on what's the general pattern in the vicinity of that DNA. And so this is like a first order Markov model, where all it does is, by first order, it says, does it matter what the preceding nucleotide was, right? If I have a C, am I more likely to get a C right after that? And in, say, mammal genomes, for example, if you have a C, you're much more likely to get a G right after it than any other letter, because CG is a methylation site. So especially around gene regulatory regions, you will get an overrepresentation of CG, okay? And so Markov models allow you to build that into your expectation for, if the DNA sequence was random, it would generate this pattern, okay? So just for people that are interested, this is what a first order Markov model would look like. And so this pattern on the left is showing the arrows, like the A to T transitions and the T to A transitions, the A to A. That's what all these little loops are. And then you translate that into this table, right? That's the probability of going from any nucleotide, from any of the nucleotides into one of the other nucleotides. And they all have a separate probability, which makes the expectation look a little gnarlier, but conceptually, it's not that complicated. And these Markov models can get very deep. So this is the sort of simplest case where it only takes into account the previous nucleotide. You could extend that to any dimensionality that you thought was biologically relevant. Okay? So for example, if you knew you were in a gene region, you could have one Markov state where it says, oh, I'm much more likely to get C's and G's in this region. But if I'm in an intergenic region, I'll use this other model that says I have equal probability of going A, C, G, or T. Okay? So all of that is to say, there are multiple ways you could come up with a null expectation for the probability of seeing any particular k-mer. So going back to our real example, is it statistically surprising to find a 9-mer that appears 3 or more times within this 500 nucleotides? So we're going to just use that simple multinomial model to see what's the expectation there. Okay, so we have a sequence of length n. In this case, it's 500. Our k-mer length is 9. Our alphabet, all equally likely. And the probability that you see a given k-mer, we already said it's 0.25 to however many nucleotides you have. In this case, the k would be 9, right? How many possible windows are there? That is, if you slid along our 500 base pairs, you can only do that in chunks of 9 so many times, right? So you've got to adjust the number of windows based on your pattern length. So, and then what's the expected number of times. So this is like the average number of times you would see a particular 9-mer. If you crunch through that multinomial model, what you will see for K9 and N500 is that you expect to see any 9-mer 0.001877 times, so way less than one time. And so intuitively, think about why is it so rare to see any particular 9-mer? Well, that's because you're talking about a specific 9-mer. So like a string of 9 C's in a row, very unlikely because you had to, like, probability of C, 0.25. Another C times 0.25. Another C times 0.25. That number keeps going down and down and down the longer that K-mer gets. Okay? So bigger K-mers are less likely to occur by chance, a specific K-mer. And so if you look at the distribution down there in the right, I know it's kind of small, but you'll see like the probability of any particular K-mer is how many times do you expect to see it in a 500 nucleotide sequence? Well, you expect to see it about 0 times on average. Okay, you can barely see this tail, right? Going out to, it only goes out to 6 in this case, but that's just summing up all those probabilities that you might get a specific K-mer. Okay, and this distribution, I won't get into necessarily the logic of why you would sample from a binomial or a Poisson, other than to say a Poisson distribution for a very rare event is just a better, faster approximation than actually doing the full binomial. It's not worth digging into that in case if that's maybe, hopefully, at least for some of the data science people, ringing a bell for probability. So under the naive multinomial, any particular 9-mer is expected to occur way less than one time. So these two K-mers are both really highly overrepresented. So again, backing up, see the forest, not just the trees. We're looking for a hidden message. These patterns are way overrepresented in this region. So maybe that's our hidden message. Do you notice anything else about those two patterns? Yeah, they're reverse complements of each other. So really, this is one pattern. It's just on the opposite strand of the sequence. And so having it 6 times in 500 base pairs is like, that wouldn't happen by chance. Okay, so this is a hidden message. So that's your candidate binding site in Vibrio for, right, this is probably where the polymerase, what it is recognizing to say start replication here. In some sense, we've solved the ORIC problem for Vibrio just using this frequency approach. But back to the problem that I hinted at earlier, if you look at the region where we know replication starts in Thermatoga, that pattern isn't there. So the binding site doesn't seem to be there. If we reapply the frequent word problem, so we just go back and look for K-mers again, you will find, 6 K-mers that appear at an unlikely frequency. I forget if they're all greater than, no, they're all 3 except for the green one. So what that means is that different genomes have different hidden messages. If you stare at those long enough, what you'll see is they are reverse complements of each other and shifted by 1 base pair. So again, there is a really strong overrepresentation of a particular pattern in here. And our algorithm missed one that is a reverse complement because it only happened 2 times, and our algorithm only spits back the most frequent. Okay? So all of that is to say, even in this new genome, we could use a similar approach, this frequency-based method, to find our hidden messages. We just have to be able to code in some flexibility in how we count. Okay, so we need to be able to count the reverse complements. We need to have it be off by 1 base pair, or the way we're going to frame it is it can have a mismatch. Okay. So that problem is sort of solved if we know where the ORIC is. Okay, and so we've limited it so far to small regions of the genome, like 500 base pairs. And that's sort of cheating in a way, because most of the time when you sequence a new genome, you don't have any idea where the ORIC is. So if you take our back of the envelope computation that said finding a 9-mer 3 times is way unlikely, but you expand that to the Vibrio genome, which is about 4 million base pairs long. So now our K is 9, but instead of being 500, our N is 4 million. Now we expect to see about 15 of those 9-mers just randomly. Okay? So if we had to account for the whole genome, our distribution becomes non-zero. So how do you decide, if you have to look at the frequency in 4 million, what is too frequent? Like, so what is a hidden message? Well, a useful approach is to chop up the genome into 500 base pair chunks and say, are there overrepresented K-mers clustered or clumped? Okay, so can we find clumps of overrepresented K-mers? So that original strategy was, if we know we've got a small region, that is the replication origin, look for the message in there by finding frequent words. Now what we're saying is, well, we could use that strategy and kind of flip it around by scanning across the genome in little pieces and see if there's a little piece that has an overrepresented message. Okay? And then that little piece that has an overrepresented message is our candidate for the ORIC region. Okay. So how do you define clumps? This is problem number 12. We'll probably not get to this one, but I'm going to give you a strategy for solving these. This is, once you've got the algorithm for number 10, sliding windows, this is taking a window, sliding through that window, then moving the window, sliding the window. So it's like a nested sliding window. And then obviously there's a bunch of bookkeeping that has to happen there because you're looking for the clump that has the greatest overrepresentation. But this is building more sophisticated code from pieces that you've already learned. If you take that algorithm and apply it to something like the E. coli genome, what you find is that there are over 1,000 different 9-mers that form clumps. So some of you that were having a hard time thinking, oh, that's circular. Yeah, it's a little circular because which of those clumps is actually having the hidden message that we're looking for, not at all clear. Okay. So which of those 1,000 different 9-mers that happen in clumps in the E. coli genome is the ORIC region? Well, at this point, if you're a pure computer science person, you may just give up because that's not a computationally addressable question. But if you're a bioinformatician, you say to your microbiologist friend, hey, can we go have coffee again? There's a theme here. All science runs on coffee. But teach me about replication. Is there any more information in how replication works I can use to refine my computational search? Okay. Well, in this case, not surprisingly, turns out there is. Right? DNA is double-stranded, so it's bidirectional. I've already hinted at this when you're tearing that circle apart. You've got two strains. They go in opposite direction. There's polarity there. But polymerase only goes in one direction. It always goes 5' to 3'. So what that means, and this is thinking back to your, maybe high school, maybe freshman genetics or sophomore genetics, I guess here, how replication works. And so the leading strands, right, as the replication fork opens, those things are being synthesized. So those strands spend very little time single-stranded. It's double-stranded because as it opens, it gets added to. It gets replicated. On the lagging strand, though, because the polymerase can't go in this direction, right? It can't go from 3 to 5. It has to wait until that fork is open wide enough to fill in the fragments. So those are, Okazaki fragments. Remember those? So it has to build this in pieces. And the reason that's important is that those strands spend a disproportionate amount of time single-stranded while they wait for the fork to open wide enough. And so why does that matter, whether they're double-stranded or single-stranded? Well, it turns out that when you're single-stranded, your mutation rate is much higher, but in particular, the deamination of C's to T's happens like 100 times more frequently when it's single-stranded. So you're basically, this cartoon down on the bottom, the thin single-stranded lines, or the part of the genome that is most often single-stranded, it's constantly losing C's by mutation to T, okay? And so on this direction, that the blue side, the forward strand, is losing C's. On the other side, right, because replication is pulling this circle apart in both directions, it's the green side that's losing C's, right? And so what happens in evolution is, how do you repair that mutation of C to T? Well, it's got to fix it so that now in this direction you've got that C turned into a T, but on the inside, like the complement now has to be an A. And over on this side, where this green C turned into a T, the blue side has turned into an A to be the complement. And so what this means is, on this half of the genome, G's are going down. And so if we looked at the difference between the number of G's and number of C's, that number is decreasing. And over on this side of the genome, that number is increasing. So thinking about, if we're scanning along this genome and we're counting the difference between the number of G's and C's, we should see it increasing along this way, and then we'll get to a point where there'll be a deflection and it'll start going down. Okay, so this is a little easier to visualize graphically. So here in these two, I've drawn the purple and the orange just to map it onto this graphic. So as we scan along the genome, our G minus C is going up, up, up. As we scan around this side, and then it goes down. And it's at that inflection point where we expect the ORIC to be, okay? So that's this inflection point right here. So our coffee with our microbiologist friend gave us some hope, because now, like, this is computational again. all I have to do is find the part of the genome that minimizes this GC skew, right? Where is that G minus C the lowest? Okay, and now that's a candidate for RIC. Hopefully it's going to overlap with one of those, Kamer regions that we found. Problem number 13 is to actually do this. You won't have to plot it, but it's a super easy thing. And in fact, if you follow the steps, ChatGPT will probably ask you if you want it to plot it, just to visualize. Right. So finding that, if, again, we go back to E. coli and we run that, we find this region that has the minimum skew, and then we find out there are no 9mers in there, so it didn't overlap. Okay, so that's how science goes sometimes. The problem is a real biological one, and that is that DNA polymerase binding is a protein-DNA interaction, and so what that means is it doesn't require strict sequence conservation. Right? So it's not a one-to-one matching. That's why at the beginning, the pattern matching strategy, I said, oh, that's a good idea. It just doesn't work. Okay. At least the sort of first pass that we've gotten to this point doesn't work. And that's because we really just don't understand this language of how protein recognizes DNA. And that's the real biological problem. So we don't know that language of protein-DNA interaction very well. The computational problem then becomes we can't count DNA sequence matches directly. Right? You can't just scan along. Even if you do the reverse complement, it's not going to tell you in this case. So again, going back to, can we do some flexible accounting in terms of how we do the mismatches in the one that I showed where it was shifted by one? That's the next step, and that's the rest of the Rosalind problems. It's, can you create code that counts all the Kamers, including their reverse complements, and allows for up to D mismatches? So if I say, it's okay if it's a 9mer and it can mismatch in 3 spots, any 3 spots. Now count the Kamers and see if there's anything overrepresented. If you do that for E. coli, you find some super overrepresented patterns. And so Hamming distance, just introducing that term, that is what D is. If you have 2 sequences that are the same length, D is just the sum of how many mismatched sites there are. So the last 5 problems here lead you to how do you do that? How do you count the mismatches? How do you generate then all the Kamers you're going to need to count? Okay, if you're going to allow mismatches, that number of Kamers that you need to look at gets really big. And just to give you an idea of how complex this really is, some of the Kamers that you need to count don't actually exist in the genome, but they're a few steps away from something that exists in the genome, and so you need to still account for them. Okay, so the next step will be, how do you count all the Kamers that are within D mismatches? And then really those last 2 are the solution, and then the solution counting all the reverse complements. Okay, if you took this class last year, this would be the next 2 or 3 weeks. This is until next Monday. And so how do you get there? Well, ChatGPT is your tutor. So let's go to Rosalind number 10. So you should see a bunch of new Rosalind problems that popped up this morning. And you will note that there's no, like the deadline is just the end of the class, so the end of the semester. Having these completed is not your homework. Okay, just to be clear, the shift is in the what I want you to get out of this is not learning to code. It's learning to translate biological problems into computational problems. So I expect you to solve these with ChatGPT. Okay, and what I'm giving you in these slides are the prompts to help teach you how to do that in a way that informs translating a biological problem into a computational problem. Okay, so our first prompt, okay, I'm just going to copy paste. Let me open up ChatGPT. And, oh, that's tiny. So I copy that. Oh, I have too many windows. Okay, that should say paste problem. So we'll go to Rosalind, number 10. And this is what I mean by paste the problem. So what you're given, what you're going to return. If you haven't noticed, shift return in Rosalind will let you skip down to the next line. You don't have to do that. It's pretty smart. But that way you can put stuff on, you can organize the prompt without actually submitting. Because if you just hit return, it will submit the prompt. Okay. So now I'll send that. Let it think. And you'll get something that hopefully makes sense to you. And the first problem isn't that complicated. It's sort of intuitive. We're given a long string of text, a long DNA string, and a short one that's the pattern. Our task is to count how many times the pattern occurs in the text. And then it sort of unpacks the expected inputs, the expected outputs, and then gives you a conceptual idea of what algorithm you might use. A lot of time, I'm not sure, I don't always get this performance report, something that we aren't going to spend a lot of time on yet. When we start talking about multiple sequence alignment, we'll talk about computational efficiency, something called big O notation. But this is like, does it scale linearly with time or exponentially with time? You want to get as close to linear with time as possible, because exponential with time means it might take a really long time. Okay, so next prompt, right? We've got our straightforward English interpretation of the problem. Whoops. Now I'm going to ask it for pseudocode. Now, for those of you who are really interested in learning to code, this is where I would not just jump to the next prompt, which is going to be ask it to give you the code. I would try to develop my own solution. So try to translate this pseudocode, which is giving you the outline for what it's going to tell you that's runnable in Colab. Think about, from the Python that you know, how would you code this? And if If you're just starting out, that probably requires a lot of going back to the tutorial from last week. And how do I code a for loop again? Okay. Now we're going to get the Colab. So now we're going to ask it to give us Python code that we can run in Colab. Now ChatGPT can solve this problem for you. There's not a need for us to paste it into Colab, but I think it's a valuable exercise, and we're going to try to help build some good habits in terms of doing git commit and all that kind of stuff to hopefully train you to do this in a reproducible way. So it spit out the code, wrote a function to count patterns in the text, and it even, down at the bottom, gave us example usage for Colab. You could put all this in. Actually, we'll just go ahead. What happens if we put all this into Colab? Start a new notebook. I'm going to name this module 4, just to keep it straight. And so I just pasted in exactly what ChatGPT gave me, and see what happens if I run it. I don't actually know. So it took a second, probably because it was connecting to the resources for Colab still, but it says number of occurrences 2. If we go back to, if we go back to Rosalind, yep. Control V or Command V. I did. So when I do copy paste, if you're on Windows, it's Control C and Control V. Command Mac, it's Command C, Command V. Also, just as a note, if you do Control or Command Shift V, it will paste as plain text. Where you see it, occasionally it pops up with what it looks like it's trying to paste a picture in there. It's because I forgot to hold the Shift key down. It doesn't matter. You can always just delete that thing. It actually doesn't matter if you leave it in there, but yeah, so I'm just doing Command C, Command V. I think you can right clicks.
That doesn't seem to work. I don't seem to be able to copy and paste it into the lab. I was having to put it into the Gemini AI box, and I asked it to run the code. And when I Googled it, it told me that I couldn't copy and paste it into the lab.
That's weird. That's definitely not true.
Very weird to me.
We can yeah, if Zoe can't, we'll fix that in a minute. Okay, so that was the code. We ran it in Colab. Now, step 4 is verify with test data. Now, mine ran because ChatGPT in its output gave test data. And coincidentally, it's exactly the test data for Rosalind, right? So you can see people have done this a lot, and like enough that ChatGPT pulled it from the training data in the internet. So let's because that's not super useful. It's not going to be able to do that. Well, I shouldn't say that. I don't know if it will do that for all of the. Actually, that's a sidetrack. But you can see where it got the, like, why did it give us our test data exactly? Or our test data, our sample data set, right? Because those inputs from Rosalind's sample data were in the code that ChatGPT gave us. So step 5 is this, or 4 is verifying with test code. So we kind of just did that, right? We got the right answer for the sample data. Practicing generalization is the next step of that. So we can, okay, this is a very helpful step in terms of learning how. I was about to get ahead of myself with the debug data sets, but let's do it again. Okay, so we're going to ask ChatGPT to modify the code so that it will run in Colab on a file that's downloaded from Rosalind, right? Because when you run a Rosalind problem, you ask it for data, it downloads it to your computer, right? So this is a handy step if we know how to modify the code to just get that file into Colab. Okay, so this gave me more complicated code for sure than what I would have done if I were coding this myself. That's okay. Gonna add a code cell here. Paste that in. And then what happens if I run that? It says choose your Rosalind input file. Well, I haven't gotten a Rosalind input file yet because the sample data was just coded into the original code that I pasted. So I'm going to download this data set. So I clicked download, and it's downloaded this for the 3rd time into my download folder. So I'm going back to Colab, click choose files, go to my download folder, click that file that just, and so as soon as I double click that file, it uploaded it to Colab, ran the code, and then gave me the count. So it gave me a lot more than what I really need because it's saying, okay, it read this file. The text length in that file was 946. The pattern length was a 9mer, okay? And then the count of that pattern is 23. So I'm going to go back to 20 here, type in 23, submit. Congratulations, we solved the problem, okay? Now, that if you ended there and didn't understand like what just happened, that's not where I hope you'll be, and it will get harder and harder as the code gets more and more sophisticated. So I, having sort of understanding the whole problem, was able to interpret this text that it was giving me, but there's a whole bunch of code here like it has a regular expression in there for splitting the text file. So this is where you could ask Gemini to explain the code. But that gets you the solution. Practicing generalization is where I was skipping ahead to a minute ago. So what if that had been wrong? Or what if you want to test it on, debug data sets? So this is probably going to vary from system to system, but if I click that button, nothing happens because all my browser pop-up blockers stop it from actually downloading. So what I have to do on my computer is right-click and do Save Link As. And then I can save it as that. And even then, it doesn't download it. It starts to, and then asks me if I want to keep it because it says it's an insecure download and that it's being blocked. But if I hit keep, okay, then in my downloads folder, I end up with a zip file, and I have multiple copies of it again. In that zip file, I get test data. So there are 6 text files here. So I could rerun that code that I just ran, but tell it to read this text file. And then I should expect to get, that output. So let me go ahead and show you what rather than just say it, do it. All right, so I'm going to rerun this code in Colab. I'm going to choose a file, input file 2. So it read that in, and my count at the end is 2. So look at my test outputs. Output 2. Oops. So output for number 2 is 2. So it got that one right. Okay. All of the tests, so the debug data sets are usually meant to find edge cases to make sure that your thing is running correctly. Like the most common way to get this wrong is for your sliding window to not be shifting correctly. Like it'll run off the end, or it doesn't read the last chunk. Okay. And then the last one is the sort of tutoring one. And so these are typically kind of mental exercises to see whether you understand the logic of the algorithm. That's usually what it's going to spit back. So here it's explaining, right, how the sliding window works and how it's counting. Okay. Questions? Yeah.
How do you choose files? Does that just pop up?
Yep. So where is the code? Yeah, so this little code block, I don't know if you can see that. That's what tells Google Colab to pop up that choose file thing. And I should also say, like, from Google Colab import files, that is that function right there. So this files And how do I know that? Well, just from playing with Colab and saying, what's the most efficient way for me to get a file uploaded from my computer? Um, and so it gave me just the code, right? So now, like if you open a lot of my Colab notebooks, they start with that because the first thing I always want to do is have it ask me what file do I want to work on. So it will just have those two lines.
That's how I found out how to do enter text instead of just having to manually edit the code itself all the time. Because now I just learned how to use underscore text within it.
Yep, the input command, right? I think we did do that one. I had that one in the notes. I did not have this one explicitly because hopefully you learn it doing this. Other questions? Okay, so definitely try to get started so that we can work through any potential obstacles on Thursday. On Thursday, I will not talk the whole time. Promise. Uh, we're going to take transcription factor binding to the next level and say, well, now I don't just care about the RIC. I have a genome file, and I have the annotations for that tell me where the genes are. Now take that annotation file, pull every 1,000 base pairs upstream of a gene, and tell me if there's anything overrepresented there. Or if I have a specific transcription factor binding site, find it within some certain parameters for mismatch. Okay? So that way we can try to find all the genes that are co-regulated just based on they share the same transcription factor binding. Okay? All right. See you Thursday.